35, 30, 27.5. Female, stage 5. 36, 29.5.
They save the mature males in cool containers. With the fully ripe females, they immediately begin extracting the eggs by pressing lightly on their abdomens. Sperms from the males are obtained using the same method. The female produces between 300,000 and 500,000 eggs per year, laying between 80,000 and 100,000 eggs at a time, 4 or 5 times during the spawning season. The quantity varies depending on the size and age of the fish, with larger fish generally laying more eggs. The fertilized eggs are left for a short time, then put in plastic bags of seawater. Oxygen is pumped into the bags, then they are tied and covered with a wet cloth to ensure that the water temperature does not rise. It is essential to complete this process quickly to obtain viable eggs, since the temperature and quality of the water affect survival and hatching rates.
الو مرحبا 5 دقائق يكون بالدائرة.
Upon arrival at the laboratory, the fertilized eggs are washed, cleaned of debris, and put into graduated cylinders for separation of the good fertilized eggs from the bad ones. The good eggs rise to the surface, while the bad ones sink to the bottom. Then the number of good eggs is estimated and are put in hatching trays through which clean water is passed. Silver pomfret eggs are spherical with a diameter of 1 millimeter. The eggs float on the surface of the sea, and often it is possible to clearly distinguish the oil globule inside the fertilized egg. Water temperature plays an important role during the egg's incubation period, which lasts about 22 hours in water at 27 degrees Celsius and 15 hours in water at 30 degrees Celsius. The development of the embryo is followed under a microscope to determine the percent of the eggs with embryos that are forming normally. After 40 minutes at 29 degrees Celsius, the cytoplasm inside the egg begins to divide into 2 cells, then into 4 cells, then 8, and so on. After 145 minutes, or just under 3 hours, the number of cells multiplies until a cluster of cells has formed. This is called the morula stage. After that, the cells begin to separate from the yolk and form in the upper region, which is called the plastoderm. The formation of the embryo begins after 6 hours, at which time the spinal column and eye sockets become visible. The embryo takes shape and fills half of the egg after 9 hours, and we are able to see the division of the muscles. The pigmentation of the eyes can be distinguished after 11 hours. After 12 hours, the details of the tail appear, and the heart begins to beat. After approximately 15 hours, the embryo begins trying to break out of the egg. The embryo then finally breaks out of the egg and swims away with a wave-like motion. This begins the larval stage. After they hatch, the larvae are counted and transferred to 1,000-liter fiberglass tanks. Larvae lives on its yolk for 3 days before its mouth opens and its eyesight is strengthened. The best potential for larval survival in tanks occurs at a density of 30 larvae per liter of water. Green algae, on average 200,000 to 300,000 cells per milliliter, is added to the culture tanks where the larvae are being raised to improve the quality of the seawater and to even the distribution of the larvae throughout the tank. Live food plays an important role in the life of the fish larvae. Therefore, the larvae are fed live microorganisms 250 microns in size, called rotifers. The circular movement of the rotifers attracts the larvae, which can pick them up easily and quickly by the 3rd day after hatching. Rotifers are considered internationally to be the best live food for marine cultured fish larvae. Five to 10 rotifers per milliliter are added daily for feeding the larvae until the 30th day after hatching. The process of raising rotifers in mass quantity is completed in the department laboratory, where they are fed on different species of green algae to raise their quality and to ensure that they contain essential, highly unsaturated fatty acids, which are important for the growth and survival of silver pomfret larvae. The live food production task produces 2 types of live microorganisms. The first type is a unicellular phytoplankton, which is used as food to produce the second type of microorganism. The latter is a multicellular zooplankton called the rotifers. During spawning season, we can produce up to 10 tons of different phytoplankton species, plus 2 billion rotifers per day.